So while you do that, Angels, I'll just do a brief introduction. So Angels is a professor at UC San Diego, and she was the person selected in our process to choose someone to develop a more complex model in a dish, which in this case is organoids for KS. And she's going to be using the lines that we have created to build those new models and type those new models and understand what the phenotype of organoids, which then completely opens up, as like Yael mentioned earlier in her talk, organoids are now kind of an accepted method that you can use to validate potential therapies. And so when we looked at the spectrum of what was available in KS, there was sort of a glaring piece missing, which was that organoid piece. And so we've decided to fill that by funding this work. And so very excited. She's going to speak about obviously the stuff she's done in the past, and she's just getting started with our lines now. So, but Angel, I'll let you further introduce yourself and take it away. Thanks.
Oh, okay. Thank you so much for the introduction and for inviting me. It's a pleasure to be part of this community. It's an honor. And I'm gonna, do you see my slides? They are fine. So
yes, slides are coming through perfectly.
Okay. As Eric mentioned, I'm a recent appointed assistant professor at UCSD, and my lab is embedded in Dr. Motri lab, who is a pioneer in brain organoids. And I'm gonna tell you today a little bit of what we are starting to do, but as Eric mentioned, we are starting experiments. So I don't have really meaningful data to share yet, but I want to tell you a little bit of what the importance of organoids in understanding neurodevelopmental disorders like the Kleefstra syndrome. Because everyone in this room probably agrees with me, the human brain is one of the more complex organs in the body. It's formed by billions of cells that are precisely interconnected, and that is a blessing and it's a curse. Thanks to this, we can have these great abilities, form memories, control our movements, but this process to have all these billions of cells orchestrated together to work, it takes decades to develop, which makes the system very highly vulnerable. And it has, as you know, are you aware, like just dysfunction in hundreds of genes, and even environmental factors during human brain development can disrupt this proper development and resulting in disease. So we have how do we study disease? We have a challenge to study the brain, the human brain, because we cannot have accessibility to living human brain. What I mean by this is that we cannot study the same brain at different stages of development and manipulate pathways and to study how it reacts. We are limited to study postmortem tissues, which can provide a lot of information, but it's a snapshot in the development of the human brain and not a continuum spectrum of things happening. We can also have some information, some window into what's going on into the brain through blood or even cerebral spinal fluid, but those are indirect measurements where they can be very useful for biomarkers of disease, not to really understand how the cellular molecular at the cellular level things are changing and being disrupted. We can also have imaging, like the living brain, living human brain, but the resolution is not there to really see what's happening inside the cells and in different cell types. So how do we study this? Most of the studies have used mouse models, which have been very useful to understand a lot of the mechanisms behind the human brain development, but they are not humans. The brain of a mouse and the brain of a human is very different. So as Neil mentioned this morning, we were very lucky to have James Thompson in 1998 and discovering how we can generate human embryonic stem cells from zygotes. And these cells can differentiate to any cell of type. But even more relevant, in 2017, Shinya Yamanaka discovered the reprogramming factors where now from somatic cells from individuals, we can actually generate these induced pluripotent stem cells that behave like these human embryonic stem cells, and we can manipulate them and become transforming any cell that we are interested in studying. This is very powerful because allows to have like patient-specific phenotypes as the genome of the patients can be maintained in these cells. There is a lot of research that has been done in what we call two-dimensional cultures, which are like here on the left, for instance, we have iPSC that have been transformed into neuronal progenitor cells, which are the beginning, what will generate neurons later. Here on the right, we have a picture of neurons in culture, and many different diseases, neurological disorders, not just neurodevelopmental disorders, but also neurodegenerative disorders have been modeled with this type of cells, and we have learned a lot. Even Kleefstra syndrome has been modeled by these two-dimensional systems. As Neil also mentioned this morning, there are a couple of papers where they use actually patient-derived neurons, and I show here the title and the mutations that they had. This is the EHNT1 gene that you are very familiar with. It's really large, has a lot of domains, and in this paper they used these patient-derived lines, and they found confirm that these cells had lower levels of EHNT1, and that's one of the reasons why Kleefstra syndrome is considered like an haploinsufficient condition. And they found some neurological and neuronal morphological changes, altered neuronal activity, and also changes in transcriptional pathways. Same with one of the Niels paper, where by using, he converted the iPSC cells from patients into excitatory cortical neurons, mixing with rodent astrocytes to increase the neuronal, to enhance the neuronal network, and also show lower levels of EHNT1, and also show some phenotypes in the neuronal network from the formation. But two-dimensional cultures, like the ones shown here, they really lack the tissue complexity. They also don't reach the level of maturity to really replicate what's going on in the brain. They are also homogeneous, which is really homogeneous, meaning there is usually one population of cells, although you can do co-cultures of different cells, as Neil mentioned this morning. But you add them, it doesn't follow the normal progression of human brain development. And it's great to find specific phenotypes that happens in one type of cell. But as a brain, we want to understand how everything works together in this orchestrated and complicated, complex soup of cells that is the brain. And also another limitation of these cultures is that you can only maintain them for so long in culture. If you're really, really careful, maybe like 1 month, couple of months. So it also limits the maturation, how mature we can make these cultures. So we, again, were very lucky in 2008, Dr. Sasai's lab. What he discovered is that these induced pluripotent stem cells, if he puts them in instead of being attached into a culture dish, floating and moving, he was able to show that they generate what we call cortical organoids, cortical brain organoids. They pretty much, these cells, they self-aggregate and self-pattern each other. It's the self-pattern. And if we cut, so these cortical organoids, I'll show you a picture of a real one later. They're about, usually in the lab, they're about 2 millimeter to 5 millimeter big, large. And when we open one of them and then we use techniques like immunostaining from different markers that we know exist in the cortex, we can see how actually the cells inside these organoids, they kind of pattern each other in different layers, like mimicking what happened in the layers of the cortex. So that was very exciting in the field, and it has been an explosion. A lot of labs now can generate these cortical organoids, and it mimics early stages of development, as I show here. This is just simply so you understand what we actually can recapitulate with organoids. Pretty much what we are doing is mimicking early stages in embryogenesis when the neural tube actually forms. So you have this neuroepithelium that falls in, and then it forms this neural tube that it will, when it closes, and then the frontal part of this tube expands. This is what will be the telencephalon, the cerebrum. And if you cut a section here, you'll see what we call the cortex. This is a really early, like few weeks during development, and this will be what will give rise to the spinal cord, right? So if we do like a staining here again, it will be similar to what we see in these cortical organoids when we do like a section, like a coronal section in this organoid. You see these little circular structures, which actually are mimicking this neural tube that we see during development. These are the early progenitors, neuron progenitors that will give rise to all the neurons that you have in the cortex. So this is what we are mimicking with cortical organoids. And there are many different labs, as I mentioned, working on this, and every lab has kind of like their own recipe. In the Motri lab, this is the recipe that we use normally. We start with, I mentioned iPSCs, and then we put them in a shaker, in the incubator, so they are moving. And we have followed different stages by changing the media and adding different factors. I will go a little bit into that also. And we start making these little balls that they grow over time until we get to the maturation, and we can keep these cortical organoids for a long time in culture, for months and even years. And this is just to show you how we culture these organoids in the lab. They are in these plates. There are these little balls here. These are kind of like in the small side, but you can see how they look. This is in the plate. We can do also staining across the whole organoid, so you can see how different proteins distribute around the inside, and you can see what I was mentioning before, these structures that they recall like the neural tube that we call rosette in the cortical organoids. So what things that we can do with these cortical organoids is also record neuronal activity. And Neil also mentioned actually something that is going to be a little bit repetitive, but we have these dishes that have at the bottom of the dish, they have these electrodes, that these are these little black dots here. And we can add the organoid, attach the organoid in these electrodes, And then we can actually measure how they fire. So you can see here on the right, when they fire spontaneously, we can also stimulate, but this is just a spontaneous activity. And then we have ways to measure how each event is here marked, or if there is like a series of events that is called bursts appears here, and sometimes we see synchronization across the electrodes. These are different electrodes which we interpret as the neuronal network, as you can see here, how these neurons come out from the organoid. They connect to each other and they talk to each other. So we have ways to measure that. And in the Mutri lab, one of the papers that they published was to actually record this neuronal activity for several months, like recording every week, and then analyzing how the pattern of this neuronal activity was. And as you can see, just without going into details, is that young organoids, they kind of like have a simplified network response. There's very repetitive, always happens at the same time. So you have this peak, and the more this peak here, and then the more they grow, the more complex the response occurs. And when they saw this complexity, these oscillations, it kind of remind them of the EEGs. So what they decided to do is to take EEGs from like hundreds of neonates, premature neonates that were available, and using AI, they were able to train this the interface to predict the age of the pre-neonate, right? The premature kid, baby, and then what they did is they feed into this AI signature. They feed the neuronal activity that was recorded in neuronal activities and ask whether they could actually predict the age of the organoid by using the EEG features that they feed into this AI. And in this graph here, this it's amazing. As you can see here on the blue side on the graph, they were able to, by just feeding in the neuronal activity that they recorded, they were able to predict the age of the organoids to a certain extent. So that was an important finding. Then what else can we do with these organoids? Many different labs are use them to model disease and to test for therapies to find new therapeutic targets. And how we basically do this is we usually get skin biopsies or blood cells from what we call neurotypical, which is individuals with no diagnosed condition, or from affected individuals. We can generate the induced pluripotent stem cells, and then from there we can generate, in this case, cortical organoids. But these days, actually, the field has really expanded, and you can generate organoids that recapitulate different regions of the brain. I'm going to focus on cortical organoids. We can also apply genome editing to introduce or to rescue the mutations. And then our goal is to find specific disease-specific endophenotypes, and we have a battery of different techniques to assess different phenotypes. We can measure the size of organoids. We can do single-cell RNA-seq or proteomics to find specifically what's the effect in the specific cells. We can look at how the cytoarchitecture of the organoid, if that's affected by that particular mutation. Same thing with the morphology of the neurons, the number of synapses, and as I mentioned, the neuronal activity is also a nice phenotype to look for. And then what we then once we have a robust phenotype that is specific of the affected organoids from patient-derived organoids, then we can look for either small molecules or gene therapy or antisense oligonucleotides that when applied to this will rescue the phenotypes. And that's what the platform that we use. And as an example, I wanted to, it's funny that Yael also mentioned this, the Pete Hopkins story, because I think it's an amazing story. So my colleague Fabio Papas, in collaboration with Dr. Motri, they published this paper in 2022, as Yael mentioned this morning, and to use cortical organoids to study Pete Hopkins, which you know that is linked to a loss of function of this transcription factor. This is just some characteristics of these the kids with this condition. And then just to show you like how we go about to try to find phenotypes for some cortical organoids derived from patients. So the first thing he looked at was to confirm that the levels of this protein were reduced in the Pete Hopkins organoids. He also looked at different cells, not only organoids. Then he looked at the size of the organoids and found that the Pete Hopkins organoids were smaller in size. And then he looked at the cytoarchitecture that I was telling you about. So in the control organoids, you see this, what we call the rosettes. Remember that they are like the neural tube, mirroring the neural tube. It's very clear the cells organized around these rosettes, but in contrast, in the Pete Hopkins organoids, he was never able to see these structures. And that was correlated with a reduce in the proliferation of these cells here, the green cells that are surrounding these rosettes, which was also recapitulated in post-mortem of Pete Hopkins individual. So this model was really good in recapitulating what was shown in an individual. Then he decided how to rescue this. Well, the obvious idea was, okay, if I can now increase the levels of TCF4 to normal levels, would that be enough to rescue the phenotypes? So he designed a very clever approach using adenovirus to just have the correct amount of TCF4 expressed and no more. And as you can see here, these are the control organoids. Remember, these are the Pete Hopkins organoids, which are really not organized. And just by adding this AAV with TCF4, it was able to rescue the cytoarchitecture of these organoids. And then same thing, he put these organoids in these multi-electrode arrays that I was telling you about, and was able to correct, as you can see here, these red lines are kind of like representing the maturity of the neuronal network in the control organoids. That was very reduced in the Pete Hopkins organoids, but then when adding the AAV, this was restored to almost control levels. And as Yael was mentioning, they got a grant from CERN to take this to the clinic. I just learned today that has been IND accepted, so that's amazing. This is 2025. The paper was published in 2022, so it shows that when you have the right group of people, the village, it takes a village to take this to clinic. So I'm really excited to hear this. It's getting into clinic. And so as you can see, brain organoids can be used to maybe speed up like like in this case, to use them as a platform to look for potential therapies and test them. And especially now with this FDA Modernization Act in 2022 that allows the application for market approval for new drugs to use alternatives to animal testing. Although, of course, there are things that we can never substitute an animal, like a behavior or something in a systemic system, but it seems that human organoids can be used as to promote like the study of potential therapeutics. And NIH recently this year has released this note saying that it's going to prioritize human-based research technology. So we are in a good space for this. And then my story, how I got into Kleefstra syndrome, is that I got aware of the, as Eric was mentioning, the iDefine had this request for applications. At the time, I was not familiar with the Kleefstra syndrome, and I started reading about it. And I know there is no cure, and these kids have long life disabilities. And I wondered if leveraging our knowledge from cortical organoids, if we could contribute and help to find therapeutic models and establish the phenotype that could help us to find these therapies. So I applied, and I was fortunate to get the award. It's a pilot award to get preliminary data, so we can apply even to larger grants to continue with the research. And as you know, EHMT1, which is this gene, is it's large, has a lot of different domains that are important for the function of the protein. And there are a lot of different variations on the gene that are highlighted here with these different colors and lines that has been linked to Kleefstra syndrome. What I like about the eDefine was that they already provided iPSCs that they were generated from patients, which that takes a lot of time in the lab. So having this already ready for us to work, I thought that was really, really nice to speed up the process. And they have this, and they can provide this male cell that have this mutation in this part of the gene at the beginning of the gene, and then these other female lines which are in this other part of the gene, which is expected to create truncated protein that is going to miss all these different important regions of the protein. And so this is pretty much what they provide. We have the female lines. They provide 3 different clones, which is important from the patient, which is the mother that is non-affected. Then we have also 3 different clones from the daughter that has the mutation. And then they also provide with a CRISPR-generated line where they introduce the same mutation that is in the daughter into the mother's cell. So we have 3 different combinations. And same thing with the male. We have not done got these ones yet, but we had received the female lines and have started working on them. What I can tell you is that the iPSCs, we receive 1 vial, we expand, we look at the quality of the iPSCs. As you can see in this picture, for us, these are really good quality iPS. They look really nice. We also do some QC, which is looking for genes that proteins that tell us that they are pluripotent stem cells. That they maintain the pluripotency. So we have done this with the control, the cells from the mom, the mutant cells, the Kleefstra syndrome cells, and then this one that was engineered to introduce the mutation in the control line. And this is very important to have these, what we call isogenic pairs of experimental pairs, where the only thing that is different between these cells almost is the mutation. So we can really explore what is specific for that mutation. So it's really important to have these engineered cells either to introduce the mutation in the control cells or later to also rescue the mutation in the cells from the daughter. We have these three now, and then we apply our cortical organoid protocol. Here are the different steps. We have been able to generate organoids we started with one clone from each of the conditions. So the control, the mutant, and the CRISPR engineer. And you can see this is like fresh picture from this week. They are about 30 days now, and we are growing them. Unfortunately, we only had time to start looking at the inside of these organoids, but they were really young. They're 15-day-old organoid. It gives us some idea that things are happening, but still too early to really conclude if it's going to be a cytoarchitectural defect or some reduced proliferation. Same thing with the size of the organoid. We are collecting the data. I was not able to put it on time together for this. By looking at the pictures, I'm not sure if we will see a difference in the size of the organoids. But the important thing is not just the size, but how they look in the structure, right? As I show you. So what is in progress and what we plan to do is to look at organoid size. We have the pictures. We just need to put together the data. We will plate next week. We are preparing the plates to put these organoids in there and then attach them and start recording the neuronal activity and comparing between the different conditions. We will also assess the proliferation and the correct cytoarchitecture that the cells distribute, as I mentioned. And we also will do bulk and single-cell RNA-seq experiments that will tell us pathways that might be affected. We are expecting already some from previous work in two-dimensional cultures of neurons. So my expectation is that we will see something different, but we have to look. And then single-cell RNA-seq allow us to ask whether there are changes in the neuronal populations. I'll show you an example in the next slide. We also propose to look at this histone methylation because that's one of the functions of this gene. And then, of course, it's very important to show reproducibility and robustness of the assay. So the plan is to repeat all of these in a different clone from the same genotype, from the same condition. And then we will do the same thing from the male iPSC slides. So some examples from other projects that are going on. These are unpublished data from one of the colleagues in the lab that he was looking at the control and the mutant organoids. You can see the nicely rosettes I was telling you about in the control, and the mutants seem to have some defects, not too bad, but when he quantified, you can see that the ventricular zone area, which is the pink area here, and that's represents the neuronal progenitor cells, the proliferation of neuronal progenitor cells, which is critical for developing the proper cortex, the size, and all the different cells. It was altered, and same thing with the thickness. So that's one of the things that we will do with the Kleefstra organoids. We also have, for a different project, we have the control organoids, and this is the diameter of the organoids, and these are different mutations to the same gene from this other project, and you can see how they are smaller than the control. In this case, it was very, it was interesting. There was a shared phenotype between the different mutations for this gene. Same thing here with this is an example of like an experiment of single cell RNA-seq experiment. These were 4-month-old organoids for a different project. Each little dot that you see here is a cell. And each color here is represented the different type of cells in that cortical organoid. So for instance, we have these radioglia cells, which are the cells that live here in the surrounding the neuronal tube that I was telling you. We have glutamatergic excitatory neurons, we have inhibitory neurons. So this protocol is good because it gives us the excitatory and inhibitory neurons that, as you remember Neil this morning, that's a the excitatory inhibitory balance is a very important feature in many neurodevelopmental disorders, actually. So in this particular case, we actually saw some difference. You see the green ones here, these are the control organoids and two different mutants for the same disease. And you can see how the glutamatergic or excitatory neurons are, the percentage of these cells is different than the control at the expenses of reduced proliferating cells, or radioglia cells, and we also observe changes in oligodendrocytes. So those are the type of experiments that I hope that we will be doing in the next month with the Kleefstra syndrome. And then just shifting gears a little bit, another thing that I also proposed to do in the grant is because the ED5 was interested in maybe following up with the potential neuroinflammation issues in Kleefstra syndrome. So as you are probably aware, the microglia is a very important cell in the brain. It surveils the brain. It takes care of, it helps to develop the brain properly, but also it's kind of like the cleaner of the brain. It helps to remove neuronal cell death, any potential synapses that we don't need anymore. And it is a very important cell in the brain that has not really been very studied in the Kleefstra syndrome. EHMT1 is expressed in microglia, so one wonder if there's maybe also mutations in the EHMT1 gene can also actually alter the microglia function, and that then can affect the brain development. There's one paper done in mouse models of the Kleefstra syndrome that suggests that actually, if you look at this picture here, if you are a control mouse or if you are lost one of the copies of each EHMT1, there is this pink cells stained here. There's like a change in the microglia. Either there is like an increase in the proliferation of the cells, and the reactivity of the cells. So it's kind of suggesting that maybe microglia might be affected. So we have also in the lab, we have established protocols to develop, to generate microglia from iPSCs. This is just like a picture of all the different steps that go from induced pluripotent stem cells to different stages to finally generate the microglia cells. And what do we do with these microglia cells? We can do, to study the function of the microglia cells, we can do phagocytosis assays, because that's one of the things that microglia does. They just eat up they clear the debris of the brain. So we have this assay established in the lab where we have microglia plated, and then we add to the media these particles that are bacteria particles that are linked to one fluorescent tag that only fluoresces in red when it gets into the cell. So we have this instrument in the building that is called Incucyte. That's the live imaging. So we have the cells that you can see here. We add these fluorescent bacteria particles that then the cells take up. So in this case, for this project, we have we saw, for instance, that the mutant cells actually were more efficient in phagocytosing these bacteria, and this is over time. We have the cells live, and then we just take pictures at different hours. We can use different controls to inhibit this. So we have the assay going. And that's what I plan to do with the Kleefstra microglia. We can also look at the morphology of these cells. We can stain them with different classical markers for microglia. We can look at the different shapes of morphologies of the cells, which has an indication of the state that these microglia might be. So we can also do this type of assay that can give us some clue. We can also measure what these microglia cells release into the media, which is the glutamate or reactive oxygen or this cytokine release. We can use ELISA assays to do this. And this is in the pipeline on the lab, so it will be straightforward to do. And I would like to finish by thanking to a very talented lab technician and a master student that they are contributing to help me with this project. And then, of course, to the members of the Montreal lab that it's amazing to be in this environment because they are always keeping in the latest advancement in the field. And I think that's the important thing that I define also consider when they gave me the award, that this is important to be associated with a lab that is like a pioneer in this field. And of course, I cannot thank enough the patients, the families. Without you, this cannot be possible. And I really admire all the patients and families and the strength and the tenacity, and creating these groups of collaboration is amazing, and I define, of course. That will be it. Thank you so much.
Thanks so much, Angels. And it's really cool. I think two things I want to just highlight from your talk. You said you hadn't heard of Kleefstra syndrome before we sent out the RFA, and I think that was the point. Is it takes a village, and we're trying to expand the village. So welcome to this expanding village of people who are all working on this challenge together, and you can't leave now. There's no escape.
No, I won't leave. Stay up to the end.
And I think the other thing is it's just so exciting. I know that it's early and you guys are just getting the organoids growing, but it's just so exciting to see the pictures, even though I know it's just the first wave of the work, because we had been thinking about this for so long. And so just wanted to convey how cool it is to see them growing. And also, as anybody who's on the call or who has worked in the laboratory before, sometimes things just don't work. And so seeing that it's working, it's just incredibly exciting that these cells are doing well and they're growing and you guys are getting results, and that's just, it's great. So I don't see any questions in the chat so far. I had a question about, I don't know if you guys have ever looked at, so it's interesting, the MEA type where you're putting them on the plate, these MEA plates. It's interesting because you have this, I was just kind of curious how it works for a sphere, right? You have this spherical ball that's kind of laying on the MEA. And then if you guys have ever looked at how that compares to the experiments that Neil does, for example, where he's doing MEA with the 2D culture, and do those two things tend to align, or do you tend to get pretty different results for 2D versus the sort of 3D organoid when they're, maybe even just the physical structure being different will change what you would get on MEA? I was just curious about how those two things compare, and if you guys have ever looked at that.
That's a very, very good question. And so that is one of the limitations of the system, right? We are trying to measure something that is in a 3D, in a 2-dimensional. But what we have seen and others is that what you measure at the end is what comes out of the organoid. So it becomes a 2-dimensional network, but it's much more complex than the 2-dimensional isolated neurons. So the level of activity is higher. They last much longer, and it's probably because these organoids are formed by, they come over time. You not only have neurons, but you have astrocytes, you have inhibitory neurons in there. So it becomes like a more complex network than in a 2-dimensional culture. It could be because 2-dimensional cultures maybe are, you cannot keep them that long. So maybe just like need time. But there is like a great effort in the field with bioengineers to design ways that you can actually surround the 3D structure and being able to measure the 3D structure, or even some systems, they kind of poke and punch the organoid inside. We don't like that one because it's like you are injuring the organoid, so it can affect that. And lately, just in the lab, we have published one paper that is actually really nice too, where we grow the organoids with graphene speckles, and this graphene we can activate by light, and then that generates stimulate the neurons to get neuronal activity. So that will be another way to really look at the neuronal activity in 3D. But it's definitely a challenge in the field. Yeah.
Yeah. Okay. I don't see any other questions in the chat. The last call on questions for. I did one other question while we're waiting to see if anybody else jumps in. How long can they go? Maybe you mentioned that. I don't know if I caught it, but what's the longest anybody's ever kept an organoid alive? And do they change? Are they sort of continuing to develop and really sort of rebalancing themselves as time goes on and almost becoming like a different thing by the time they get to the very end, just as a fetal tissue would during development?
Yeah, very good question. Also, you can actually keep them growing for a long time. I have some for a different project that they are like 2-year-old. We are actually analyzing now them. There is another investigator, Arlota, she's very pioneer also in the field, that she had like 5-6-year-old organoid. And yeah, I mean, the challenge is to keep them that don't get contaminated. They do change the population of cells, but you have less proliferating cells. They become like more abundant astrocytes, more inhibitory. But there is like an intrinsic barrier in these organoids in vitro that you cannot really make postnatal brain organoids yet. And it can be for many different reasons. For instance, they don't have vasculature. Right? So the center of the organoid, even though these are like tiny structures, 2 millimeters, 4 millimeters, they actually are hypoxic and they can get necrotic. So, that like over time, you only have like living cells surrounding the outside. They grow over time a little bit more. So that's a challenge also. But you can keep them for a long time. Yes.
Okay. Yeah. All right. I don't see any other questions. So I think we'll move on to the next speaker. Thanks so much, Angels.
Thank you.